This will be the first lesson on photosynthesis, and in this lesson here, we're really going to be talking about some of the properties of light, the nature of light. Before that, though, just kind of keep in mind that when we do think of photosynthesis, we do have a tendency to think about those organisms around us every day that are photosynthetic, so trees and plants and flowers. But just keep in the back of your mind that photosynthesis is also taking place in water, whether it's oceans or lakes. We have seaweed, we have aquatic plants. So the picture that we see at the left-hand side here, the multi-cellular algae where photosynthesis is taking place, and also some single-cellular organisms, protists and bacteria. Photosynthesis can also be taking place there. Plants, of course, do require light for the process of photosynthesis. And like us, plants do use a very, very specific portion of what is referred to as the electromagnetic spectrum. Electromagnetic energy is a form of energy that does travel in the form of a wave, and it's made up of an electrical component and a magnetic component, and hence the term the electromagnetic spectrum, electromagnetic radiation, or simply EMR. When we do talk about energy that's traveling in the form of EMR, it does travel in the form of a wave. So different wavelengths do have different properties. And if we do take a look at the wavelength that is identified here, it is the distance between two successive crests. So wavelengths can vary. And if we go from the left-hand side of this picture to the right-hand side, we can see that the wavelengths, they are getting shorter and shorter and shorter. As the wavelength gets shorter, the frequency or the number of these crests that we can get in a second, that is going to increase. And as we shorten the wavelength, that is also going to increase the amount of energy. So what we could then say is the wavelengths that we see at the left-hand side, the very long wavelengths like radio waves, they have very, very low energy compared to the short wavelengths that we see at the far right-hand side, which include the gamma rays. Most of this, we as humans, we cannot see with the unaided eyes. The only thing that we can really see is what is referred to as the visible spectrum. So this is the portion where we do see the different colors, ranging from the lower energy red light to the somewhat higher energy blue and purple light at the right-hand side of the visible spectrum. There are some other organisms that can see into the infrared range and that can see into the ultraviolet range. The infrared would be lower energy, the ultraviolet slightly higher energy than the visible spectrum. So that is what we see as the visible light, and it just so happens that that is also what plants see as well, if plants could see. And when I say what plants see, it's really what they are using. So plants do not use radio waves, they don't use microwaves, infrared, ultraviolet, X-rays, or gamma rays. Really what they do use is, well, just what we see, and that is the visible spectrum. In fact, what we'll see as well is plants are even more specific in terms of what they can actually use for the process of photosynthesis. So all of these different colors of the rainbow in the visible spectrum, if you mix them all together, what you do get is white light. And we can also kind of flip that around if we take a look at a source of white light, which is what we do have from our fluorescent lights or incandescent lights. What is produced by the sun as well is for the most part white light. If we do take this white light and pass it through a diffraction grating or a prism, then what we do get are all of the different colors of the rainbow. And in fact, when you see a rainbow, that is what is happening, is white light that is coming from the sun. It's being split up into its different colors by moisture, by water droplets in the air to produce the rainbow. So the reason why this rainbow is generated is that the violet light is going to be bent more than the red light, and because it's bent more, it can be split into the different colors. If we do take a look at the wavelengths that are identified at the bottom here, this is now measured in nanometers, which are billionths of a meter. Visible light ranges from about 700 nanometers for the wavelength down to about 400 nanometers for the wavelength. Again, the violet light has the shorter wavelength than the red light, so it will be of slightly higher energy. So I mentioned that plants are even more specific in terms of the light that they can use, that they can capture and they can use for the process of photosynthesis. So what we are seeing in this picture here is light that is coming in, it is striking the plant, and of course the portion of the plant that is capturing the light energy is going to be the chloroplast. And within the chloroplast, within the thylakoid discs that we see colored green in this picture, inside of the chloroplast, there are a number of different plant pigments. The primary plant pigments, as we will see, are chlorophyll pigments. And what these chlorophyll pigments do is they are going to absorb light energy. If we do take a look at this picture, though, what we do have coming in from the top is going to be white light that is made up of all of the different colors. When this white light does come in and interact with the pigments in the thylakoid discs, what we can see here is that we have some light that is reflected. The light that is reflected is green. We can also see that there is some light that passes right through. The light that passes right through or is transmitted, that is also green. If the light is reflected or if it is transmitted, that means that it is not being absorbed by the plant. Only when light is absorbed by the plant can it actually be used, and used in this case for the process of photosynthesis. So what we can see here is the light that is absorbed is going to be, for the most part, the blue light that is absorbed, and also the red light that's going to be absorbed. So what that means is that as far as photosynthesis goes, for the most part, plants are using blue light and red light from the visible spectrum for photosynthesis. They're actually not using the green light, and that's why plants appear green in color, because that is the color that is being reflected. Whatever color you see that color because that is what is being reflected. If it is being absorbed, then you're not seeing that color. If we take a look at this little bit of an experiment here, it's showing us that at the left-hand side that we do have a source of white light that's producing all of the different colors of the visible spectrum. We have a prism that is splitting this white light into all of the different colors. The top picture, they're showing that there's a little slit, and this slit is allowing only green light to pass through. And this light is then passing through a test tube that is filled with a chlorophyll solution. So the chlorophyll solution isolated from a plant. So this chlorophyll solution is going to be capturing some light energy, and it's going to be allowing other light energy to pass through. What this picture does show us is that if we do allow green light to impinge on this chlorophyll solution, what happens is that, well, it's not being absorbed. In fact, if we take a look at the galvanometer here, we can see that it's being transmitted, it's passing right through. And once again, what that means is that there is very low absorption with the green light. The green light is not being used. If we take a look at the picture at the bottom, the only difference here is now they shifted the slit a little bit, and what they're allowing is the blue light to pass through. So as the blue light passes through the slit and it interacts with the chlorophyll pigment, what we can see is now it is not being transmitted through. It's not being reflected. So what happened to it? Well, it's being absorbed. So we have a high absorption of the blue light. And once again, what that means is that this blue light can now be used by the plant, and it can be used for the process of photosynthesis. Very, very important that you understand the two graphs that I'm showing you here, that you understand what each one is. The top one is what's called the absorption spectra, and the bottom one is the action spectra or spectrum. In both cases, we're taking a look at the manipulated variable, which is on the x-axis, MV for manipulated variable. What we're doing is we're changing the wavelength of light. In this case, it is, you might notice, flipped around from the other pictures that I've showed you. We have the purple light of the shorter wavelength that is now toward the left-hand side, and we have the red light of the longer wavelength, which is toward the right-hand side. So we're changing this wavelength, and what they're measuring on the y-axis, which of course is going to be the responding variable, what is being measured is the absorption of light by the chloroplast pigment. So this isn't necessarily just the chlorophyll pigments, it's all of the pigments that you would find inside of the chloroplast. So what we can see is that they're showing three different pigments. They're showing two different chlorophyll pigments, chlorophylls A and B, and they're also showing what are called the carotenoids. So the key here is that you understand these pigments, what wavelength of light, what color of light they're absorbing. And once again, the color that they are absorbing is the light that they are able to use for various different cell processes, which of course in this case is going to be the process of photosynthesis. So what we can see here, if we take a look at the chlorophyll A, is it has a very, very high absorption at about 450, 440, 450 nanometers. If we take a look over at the right-hand side, we can also see that that same pigment, chlorophyll A, also absorbs a lot around 670 nanometers. So that corresponds to the purple or the blue light, and it corresponds to the red light. If we take a look at another chlorophyll pigment, the chlorophyll B, we can see that it again has a peak which is in the blue range, and it has another peak which is close to the red range. So for both of those chlorophyll pigments, what they are doing is they're absorbing, well, mostly in the blue area and quite a bit in the red area as well. They're absorbing that light energy. Well, what about the other wavelengths? What about in the green and the yellow? Well, they're not absorbing it. If they're absorbing it, they're reflecting it or it's being transmitted. And what that really means is that chances are the plants are not using those colors for the process of photosynthesis. So if we go down to the graph at the bottom, action spectrum, what we have on the x-axis is exactly the same. We're changing the wavelength of the light. So at the far left-hand side, it is the purple and the blue, and at the far right-hand side, it's going to be the red light. Action spectrum means something is happening, and what we're talking about here in terms of happening is photosynthesis. So the higher the peak is for the action spectrum, the higher the rate of photosynthesis, and they say that that is measured by the amount of oxygen that is produced. So what this is showing is that if you shine the blue light on the plant, you get a high rate of photosynthesis. If you shine the red light on the plant, you get a high rate of photosynthesis. Which pigments correspond to the wavelengths of light that are absorbed in the blue and the purple? Well, that's going to be the chlorophyll pigments, where we see the high absorption in the blue, and, well, the chlorophyll pigments again, where we see the high absorption in the red wavelength. So important for you to understand that, yes, it is those pigments, the chlorophyll pigments, that are responsible for absorbing light energy, and it's those pigments that are then going to be responsible also for the process of photosynthesis. What is actually going on when these pigments do absorb light energy is they're going to be exciting electrons. So this picture here is just showing a general image representing what happens when light is coming in and light is striking an element or a compound or a pigment, in this case when we are talking about photosynthesis. So it's showing light at the upper left-hand side. It's coming in, it's interacting with the electrons. So if we remember the structure of an atom, in this case it's showing the Bohr model, where we have the positively charged nucleus, we have electrons around the outside. So yes, this negative in the circle is referring to the electrons. So it is light that is interacting with the electrons. It's not interacting with the nucleus. And if the light is of the correct wavelength, this is very, very wavelength specific. So if it is of the correct wavelength, what's going to happen is that light is going to interact with the electrons and it's going to pass that energy on. Remember, energy can't be created or destroyed, only transferred from one form to another. So in this case, what we have is energy that's being transferred from the light to the energy of the electrons. It's adding energy to the electrons, and when it does that, it moves the electrons from what we call the ground state up to what we call the excited state. The electrons are excited, they've been given more energy. So these high energy electrons, now they have two different possibilities. We can simply have those electrons that fall back down again. So when those electrons fall back down from the excited state to the ground state, they have lost some of their energy. They have to have that energy going somewhere else. And usually where it goes to is it re-releases light once again. And when that happens, that's what's referred to as fluorescence. In this case, you might notice that the wavelength is longer of the light that's being released than the light that was originally coming in. And that's because with any energy transformation, some energy is always going to be also lost in the form of heat. So the wavelength of the light that's going to be released through fluorescence will be of slightly lower energy and of longer wavelength than the light that came in the first place. Having said that, that's not what happens in plants. What happens with plants is something referred to as photooxidation. Photo is referring to light. Oxidation means the loss of electrons. Not only are those electrons becoming excited, but they're becoming so excited that those electrons are going to be completely stripped away from that original atom, element, molecule, pigment in this case, and they're going to end up going somewhere else. So in this case, just in general terms, it's showing that the electron is being added to this Q, and then we end up with it picking up the negative charge and having a negative charge. So this term here, photooxidation, again, does mean that we're using light energy to cause the loss of electrons. So a little bit of terminology here. We'll just read through these points. In some circumstances, the absorption of light by a molecule will give sufficient energy to an electron to move that electron far away from the nucleus, and it will be captured by some other molecule. And a little bit later on, we'll see exactly what those molecules are going to be. So the molecule that accepts this high energy electron, it is going to be the electron acceptor. The one that gives it up is the electron donor. And the molecule that absorbs the photon will then have lost that electron. So I mentioned that term oxidation. So the term oxidized does specifically mean lose electrons. So anything that has become oxidized will have lost electrons. Whatever gains electrons is referred to as reduction. So oxidized, lose electrons, reduce or reduction is the gain of electrons. So kind of think about this as electrons having a negative charge. If you gain more electrons, the charge actually becomes more negative. So what you have done is you have reduced the charge. So that's how you can gain something, and it's still referred to as reduction. So in the case of plants, because this is all driven by light, it is referred to as the photooxidation. We're going to come across a whole bunch of reactions in photosynthesis and in cellular respiration, where what we have is something that is losing electrons, something that gains electrons, and this is just going to continue on and on and on. Gain electrons, lose electrons, gain electrons, lose electrons. And when this is taking place, these are referred to as redox reactions. The re for reduction, and the ox for the oxidation that's taking place. So all of this in plants is going to be involving plant pigments, a couple of them, of course, that we've come across already, the carotenoids and the chlorophyll pigments. So energy, as we mentioned, the key is that the energy does need to be absorbed. Pigments, that is their role, is to absorb this light energy. In plants, yes, there are many different kinds of pigments, and we're going to be doing a lab, a chromatography lab, where you will in fact see the number of different pigments, or some of the different pigments anyway, that can be present in a sample of a leaf or plant tissue. And each one of these different pigments, they are very, very specific in terms of the different regions of the electromagnetic spectrum that they are going to absorb that light energy. So the pigments that we do see, they are certain colors because of the light that they are reflecting. So if we do have plants that are green in color, those plants again are green in color because what they are doing is they are reflecting that green light, and that is usually the chlorophyll pigments. Several different kinds of chlorophyll pigments, but for the most part they are going to be reflecting the green light, and they appear green in color. That means the other colors, the blue light and the red light, that's what they're going to be absorbing, and that's what they're going to be then using to excite the electrons and used for the process of photosynthesis. So the lab that we will be doing, it is referred to as a chromatography lab, and this lab is going to be separating plant pigments, and it separates a mixture of plant pigments that we do have in the leaf based upon their differing solubilities in solution. Different solutions that we can use and different pigments are able to dissolve in or are soluble in these solutions to different degrees. So the kind of chromatography that we're using in this lab, it's referred to as paper chromatography, and it involves applying a very small drop of our sample, which is going to be coming from a leaf, onto the absorbent sheet of chromatography paper, placing a portion of the tip of that chromatography paper into a solvent, into a solution. And what's going to happen is that solution is going to slowly travel up along the length of the chromatography paper. And when it does that, if we do have any of the pigments that are soluble in the solvent, then it's going to drag those pigments along with them, and it's then going to separate the pigments based upon their differing solubility in whatever solvent we're using. So more or less, this is what it looks like. This is showing our strip of chromatography paper at the beginning here. So this is time 1 at the left-hand side. We're applying our sample of pigment solution, the plant leaf solution, toward the bottom of the chromatography paper. And then we're sticking the tip into a solvent. So this solvent or solution is then going to slowly travel upward through the chromatography paper, and any pigments that are present in the initial sample that are soluble in the solvent, they will travel along with the solvent. Some of them are more soluble than others, so we can see in the second, the third, and the fourth time here that we are separating the pigments. So we started with one original sample, which was green in color, which we applied to the bottom. After a period of time, we have the solvent that has traveled almost to the top, and this is what we call the solvent front. Some of the pigments are very, very soluble, and they travel the entire length of the solvent. So we can see here that the red one would be very soluble, the yellow not quite as soluble, and the two at the bottom would be somewhat less soluble. There may be some pigment that is left at the beginning, and these ones that didn't move at all here, they're ones that would not be soluble in the particular solvent that we're using here.